Hello guys, so here I am with my new super awesome video. So topic is DNA replication. Before that I make this kind of informative content, so please consider subscribing my channel. Okay, so good luck guys. Let's see the replication process in the simple manner. Stay connected. So as we know, in eukaryotic cell, the DNA is the basic genetic material which transfers the genetic materials from parents to offspring. So it's needed to be replicated, this genetic material. So for that, this DNA replication process takes place in eukaryotic cell. So for DNA replication, we need the basic requirements are helicases enzyme, then we have topoisomerases, then we have primase, then we have SSBs, DNA ligase, and at last, most important, polymerase enzyme, due to which the polymerization process of DNA strands takes place. So this replication of DNA process takes place in majorly three steps. It's a three-step process. First is initiation, then it's elongation, and at last there is a termination. So now we'll see this initiation process. If we consider the DNA of E. coli. E. coli is a prokaryotic cell. Here ori is the origin of replication. See, if we see about the DNA replication, there is a ori or origin of replication. In one DNA there may be one or more ori. At that point the DNA opens up. At this point DNA opens up, and it consists of, this ori sequence of E. coli consists of 245 base pairs, and this opening of DNA take place by the helicases enzyme. It breaks the bonds. Bonds are phosphodiester bonds between the strands. There are two strands: leading strand, that is from 3' to 5', and then we have a lagging strand which is from 5' to 3'. In initiation process, first the replication bubble is formed. Here we can see this is the replication bubble. This takes place. This is the replication bubble. Here we can see this is a DNA polymerase enzyme, which is the main enzyme in the DNA replication process. This is a replication bubble. This is SSBs, this is helicase, and this is topoisomerase. These are the basic enzymes which are main important in the DNA replication process. They have their important specific functions, which we'll see later. Then after we have the topoisomerase enzyme. Here topoisomerase help to release the topological stress. This stress can lead to breakage of strand. As we know, this DNA polymerase enzyme, it pushes the one strand towards other cell. So by this the stress takes place, and due to release this stress and to prevent the DNA from damage, this topoisomerase enzyme is important role. Now after all this, now the DNA polymerase enzyme, our prime enzyme, is loaded inside the replication bubble. But first RNA polymerase comes into the action to form the primer. It is primer is short RNA segment which is formed before the replication process of DNA takes place. Because DNA polymerase needs template to start replication process. As we know, the DNA polymerase enzyme needs the template, the ready-made template to start its replication process. So at last the DNA polymerase becomes active, and it starts adding base pairs by doing proofreading along with the replication process. Proofreading, its other main function is proofreading with the replication process. In proofreading, DNA polymerase checks whether it had added the right base pair or not. If it put the wrong base pair, it shows the exonuclease activity and it corrects its base pair. It also helps to remove the cleavage which takes place between the strands which are not joined by the phosphodiester bond, due to the, if A is joined with G, then no bonds are formed. So it corrects that and puts the T it removes the G and then adds T. So the AT bonds are formed, that double bond, and at GC we have a 3 triple bond. Now after the initiation process, the elongation of DNA pair take place. Here the leading strand synthesis continuously take place by the DNA polymerase. As there is a condition that DNA polymerase can synthesize only one strand continuously and other strand discontinuously. So in this, the leading strand is continuously replicated by the DNA polymerase, whereas the lagging strand is discontinuously replicated by the DNA polymerase in the short segments, which is called Okazaki fragments. This happens due to some limitations of DNA polyenzyme, as I said earlier. So in this during this process, this unpaired pairs are opened in the environment. So to protect this from rupturings or denaturing, the SSBs bind to this nucleotides. These SSBs protect the nucleotide by joining them single stranded binding proteins. SSBs are single stranded binding proteins. It protects the nucleotide from denaturing or any other activity. Now these short Okazaki fragments are joined together by the glue enzyme, which we call a DNA ligase. It ligate the base pairs which are in that Okazaki fragment. It joins the Okazaki fragment and forms the continuous strand that is from 3' to 5'. In this ligation process, the energy is used which is provided by the DNA ligase enzymes. Now the last step of DNA replication, it is termination. So it is a final step in replication. Here two replication fork of E. coli chromosome meets at a terminus region containing multiple pair of sequence called TER sequence. There are two things important in this. There is TER sequence and TUS proteins. TUS are the proteins and TER is a sequence on the DNA. So here the TUS and TER protein, this forms the complex and it arrests the replication fork and stops the replication process. So by this, the termination of replication process takes place by TUS and TER protein join together and blocks the replication bubble. So your TUS is a terminus utilization substance. It is a protein that binds to the terminator sequence. It is a protein which binds to the terminator sequence in the termination process of DNA polymerization. Now this TER is a terminal sequence which interact with the TUS protein and stops the replication. TER sequence is present on the DNA strand which is to be replicated, which undergoes the replication. It interact with the TUS protein and stops the replication. By this, which we can say the termination of DNA replication process takes place. So at last, I hope you liked my video. So please like it, subscribe my channel for more videos like this, and keep supporting, and goodbye guys.